What's the big? I mean, we want to make sure it's ready for mitosis. But what's the big thing to check? Think about it. Make sure the DNA replicated right. There it is. Exactly. And then we start mitosis. Anyone tell me some of the main things about prophase? Can anyone tell me some of the main things about prophase?
Division of nuclear material.
No. Prophase. How about the nucleus goes away? How about the nucleolus disappears? How about the chromatids tighten up into chromosomes so we can see them? How about the centrioles go to their opposite poles?
Like the microtubules, like form.
And yeah, the spindle fibers, which are basically microfilaments, microtubules. The spindle fibers form. Those are all things that happen in prophase. Centrioles go opposite poles, spindle fibers form, nucleus goes away, nucleolus goes away, chromatin tightens up into chromosomes. What happens? What's the main feature of metaphase? They line up in the center along that metaphase plate. They all line up along that metaphase plate. Now, to do that, they have to be attached to the spindle fibers. So what phase did I tell you occurs in between prophase and metaphase? And it is the attachment of the chromosomes to the spindle fibers. Prometaphase, yeah, just put the two together. That's what they did. That's what they did when they named it. It's prometaphase. It's the attachment of the chromosomes to the spindle fibers. The migrating to the center, lining up in the center, is metaphase. And metaphase is finished when they are what? Lined up. And then we have the M checkpoint. The mitosis or metaphase checkpoint, M checkpoint. And what's the main purpose of the M checkpoint? Make sure they're, well, I'm going to add something to that. You're right, but I'm going to add something to it. Make sure they're all lined up right and properly attached to the spindle fibers. Because they can be all lined up right, but if they're not properly attached, they won't separate properly. If they're not attached, they can't separate. So we need to make sure everything's cool. And that's the last checkpoint before it all happens. Now, once that checkpoint's been passed, what happens in anaphase? They move / migrate to their opposite poles. They move / migrate to their opposite poles. And when they do that, then telophase starts. And telophase is essentially the opposite of prophase. Why? What happens to the spindle fibers? They go away. What happens to the nucleus? Reforms. What happens to the nucleolus?
Becomes visible.
Reforms, reappears. What happens to the chromosomes? They loosen back up into chromatin. Essentially the polar opposite of prophase. What is the one additional feature? What do we have? Two things there. One's going on that doesn't start in telophase, and one is associated with telophase. Because at the end of telophase, what do we have? Well, hang on to that thought for a second. At the end of telophase, what do we have? Two new identical daughter cells. So in order to do that, what has to happen once all that other stuff occurs? We have to split that cell, right? Cleavage furrow. You got the cleavage furrow, the contractile ring squeezes and pinches off, and we get two new daughter cells at the end of telophase. Now, what you're alluding to starts in late anaphase, continues through telophase, and what is it called? No, cytokinesis, which is cytoplasmic division, i.e. the splitting up of the cytoplasm and the organelles. Make sense? The fluid of the organelles gets split up between the two cells. Starts late anaphase, continues through telophase. And here we're going to get a look at this, and I'm going to kind of, no, wrong thing. Don't want to quite that much. That's a little better. We start with all the chromatin, late interphase, we're all ready to go. And then we get into prophase, nucleus goes away, nucleolus goes away, chromosomes, centrioles, opposite poles, spindle fibers. Then as we get the late prophase, they're going to go to prometaphase and attach. Then they're going to line up, and in metaphase they are now lined up. Nice and neat like this, and you can kind of see the spindle fibers right there, the dark lines with the light in between, with the centrioles. God bless you. And then you see them migrate during anaphase. And in late anaphase, when they get real close, you are going to see the fact that cytokinesis is going to start. And then as we get to telophase, you see the cleavage furrow. It squeezes this all off. Nucleus reforms, nucleolus reforms, spindle fibers go away, chromatin again from chromosomes, and we have two identical daughter cells. And now they are in G1 again, and we're starting this whole thing all over again. And that's what we're going to be looking at. And here you can get a look as they start to line up. They line up again in metaphase. They migrate during anaphase. And then the get there, telophase starts with the cleavage furrow, the reformation of the nucleus, nucleolus, the disappearing of the spindle fibers, and we wind up with those two new daughter cells. Okay. That is that part of this